Welcome to Sekisui XenoTech's webinar, Comparison between the final US FDA, Japan PMDA, and EMA in vitro DDI guidance documents. Are we finally harmonized? I am pleased to have Dr. Brian Ogilvie here with us today. Brian received his Ph.D. in toxicology from the University of Kansas Medical Center. He has been with XenoTech since 1997 and currently serves as Vice President of Consulting. Brian is an author or co-author on over 50 scientific posters, peer-reviewed publications, or book chapters on the topics of drug metabolism, transport, and drug-drug interactions, and has represented the company as an invited speaker at various drug metabolism conferences. As Vice President of Scientific Consulting, Dr. Ogilvie is additionally involved in quality management and strategic planning for the company in scientifically relevant areas. It is my pleasure to present to you Dr. Brian Ogilvie.
Okay, thanks, Michael, for the introduction. And I really do appreciate everyone's time. I know that it's an unusual time with the COVID crisis going on, and most people probably staying at home, just like we are. This is coming to you from my basement, but I have the, I guess, advantage of having a nice large screen to look at while I give this presentation. So again, thanks for coming, and you'll see my title has lots of finals in it, because the question I'm asking is, are we finally harmonized? And I'll get to that toward the end. So the presentation outline, I will actually quote directly from the final 2020 FDA in vitro DDI guidance in terms of what I see as major changes. And I'll cover some of the timing strategies for in vitro studies, as well as evaluating test drugs as victims according to the FDA, PMDA, and EMA, enzymes of interest, as well as transporters, and evaluating test drugs as perpetrators according to the FDA, PMDA, and EMA. And then lastly, strategies to harmonize the design of in vitro DDI studies to meet the expectations of all three agencies. And just so everyone has a picture of the guidance documents, this one FDA finalized in January 23rd, 2020. EMA was finalized in 2013, and I know that they have been trying to work for several years on a revision. There was a concept paper that came out a few years ago, and then the PMDA finalized theirs in 2018 and released an official version, in English version, in 2019. So major changes to the final 2020 FDA in vitro DDI guidance. This is my first slide. So right off the bat, the title, if you look at the 2017 draft, it referred to in vitro metabolism and transporter-mediated drug interaction studies. The 2020 final title was in vitro drug interaction studies, cytochrome P450 enzyme and transporter-mediated drug interactions. So I originally thought from page one that this is going to be focused on P450 enzymes. So that apparent limitation in scope, really they added the following enzymes back to evaluate for victim potential if CYPs are not significantly involved. So carboxylesterases, aldehyde oxidase, I think many of us were happy to see this come back to it after it was withdrawn from the draft 2017 guidance, and then the sulfyltransferases. And then for metabolites, they are essentially the same as the draft. And I think for the most part, there hasn't been a lot of change since 2012. But the risk of DDI for active metabolites that contribute to greater than 50% of overall activity is expounded upon. And if you summarize this, it basically comes down to conduct an in vitro inhibition study of the metabolite if a metabolite is less polar than the parent drug and the AUC metabolite is greater than or equal to 25% AUC of the parent. In other words, AUC metabolite greater than 0.25 times AUC parent. And I should note that when I do not have quotes, I'm paraphrasing guidance. I'm a little bit cautious about paraphrasing guidance, so I try to be as strict as I can in terms of how I read out the guidance document. So the sponsor should conduct an in vitro inhibition study of any of the metabolites if one is more polar than the parent drug and the AUC is greater than 100% of the AUC of parent. So in other words, the AUC metabolite is greater than or equal to the AUC parent. However, the final guidance wrote this sentence that a lower cutoff value for the metabolite to parent AUC ratio may also be considered for metabolites with structural alerts for potential mechanism-based inhibition. And there are several seminal papers on what can cause structural alerts for potential mechanism-based inhibition. I would note that some authors in the past have pointed out, rightly so, that just because a compound has a furan ring on it doesn't necessarily mean that's the part of the molecule that's causing the mechanism-based inhibition. So you do have to be careful in those structure-activity relationships. So metabolites continued if, and again, this is a quote, if basic models suggest that the metabolites may have in vivo DDI liability and a static or dynamic mechanistic modeling approach, in other words, PBPK, is used for a DDI assessment of a drug, metabolites should also be incorporated in these models. The risk of a clinically relevant DDI through altered formation or elimination of metabolites should be investigated if changes in metabolite exposure levels may result in clinically meaningful alteration of efficacy or safety. So here we're not talking only about exposure, but efficacy or safety due to metabolite changes in exposure. So for active metabolites, both the in vitro receptor potency and the in vivo unbound systemic exposure, which is expressed in Miller units, of a metabolite relative to the parent drug need to be taken into consideration when evaluating the contribution of the metabolite to efficacy. So this is important because it's not as simple as it was in the past. For active metabolites, you need to know what is the potency at the receptor in question. There may be more than one. And then what is the unbound systemic exposure, and what other considerations could there be when evaluating that contribution to the overall efficacy? So there are several new phrasings, I should say, but essentially the same concept. So again, on metabolites, this again is a paraphrase. If in vitro data suggests the parent alone will not inhibit CYPs or transporters, it's still possible that metabolites may cause clinically relevant DDIs. So in this situation, the sponsor should evaluate the in vitro inhibition potential of a metabolite on CYP enzymes, taking into account the systemic exposure, again in the Miller units, and the polarity, so measured or predicted log P, the elution order on chromatogram of reverse phase high performance liquid chromatography of the metabolite relative to the parent drug, and potentially other measures. So as far as interpretation of reversible inhibition of hepatic and gut CYP enzymes, I will note that the equations for FDA now specify the unbound in vitro Ki value should be used for the R1 and the R1 gut calculations. And if you're not familiar with these calculations, I will provide tables later on that walk you through the FDA and the other agencies' equations. This is another quote. Half of the measured IC50 can be used as an estimate of Ki if the substrate concentration used is equal to its Km. And this is per Haupt, Parkinson. This is a paper that we published in 2015 in Drug Metabolism and Disposition. Interpretation of irreversible inhibition of hepatic CYP enzymes. Again, now they specify within the equation the unbound in vitro K large I, and this is Ki that is analogous to Km. So in other words, it's simply the concentration of drug that causes half of the maximal inactivation. Those values should be used for the R2 calculation. Interestingly, there's still no R2 gut calculation in the final guidance. I'm interested in this, and I know that there is actually an FDA webinar coming up this Friday with Kelly Reynolds and one other speaker. I'll be interested to hear if they cover this. I'm guessing this is not an omission, but rather there must be some rationale for this. Another quote. In the presence of inhibitory metabolites of an investigational drug, their contribution and rank order of metabolite R values should also be considered when determining what in vivo studies should be conducted. So this is something, there are equations to actually take, especially when it comes to more mechanistic dynamic models to take into account the metabolites. But this is talking about taking into account the metabolite R values and ranking them for the basic and mechanistic static models. So interpretation of CYP induction, there really is more on this than there are in some other places. So there's a minor scope clarification. It's for mRNA, only CYP3A4, not CYP3A4/5, as in the 2017 draft. And a clarification, if a result from only one donor exceeds the threshold, the drug is considered to have an induction potential. And this is basically the assumption that many of us have been working on for many years, and that you don't average the results that you get across 3 donors. You take the worst case donor that you have. A positive fold change criteria now says that if you have greater than or equal to a 2-fold concentration dependent induction at expected drug concentrations in the liver, or a greater than or equal to 20% response of positive control, that's a positive response. So the expected drug concentrations in the liver, this again is a quote directly from the final guidance, can be calculated by assuming a certain fold of the Imax unbound. So for example, 30-fold of mean unbound maximal steady state plasma concentration at therapeutic dose. And I don't know, again, the rationale for that. I think if you have certainly data on the blood to liver ratio, then you can use that in place of that, or if you know if you have some other data suggesting how much unbound drug there is in the hepatocytes. And they also define a percent of positive control is equal to, this was more or less spelled out, I mean, assumed in the past, now it's spelled out, mRNA fold increase of test drug treated cells -1 times 100 over mRNA fold increase of positive control -1. So note that the -1, that is something that's not always done, and that's the reason I put that in there. For the R3 equation, the Imax unbound is now the unbound plasma steady state, Cmax. So in the past, it was simply Cmax. This may be something of a change and allow you to look at steady state kinetics as opposed to single dose Cmax. Same cutoff at less than or equal to 0.8. The D value, if you're familiar with this equation, and I'll show you again later, can be adjusted from 1.0 if supported by prior experience with the system used. So again, on an induction, this is actually kind of a big one. No more requirement for a negative control. So for example, many of us have been using flumazenil as a negative control. And I think many of us have argued strenuously that that made no physiological sense to have something that has no binding capacity for a nuclear receptor. And it seems that the FDA has relented and taken that out. So another quote, concurrent prediction of inhibition and induction, this is my words, can once again be considered for predicting the net effect of an investigational drug that is hypothesized to be both an inhibitor and an inducer of metabolizing enzymes. However, there is concern with concurrent predictions. So if constant, and actually I want to back up on that just a little bit. So this was the idea in the guidance that appeared, I believe, in 2012. The net effect model allowed sponsors to take, especially when you had mechanism-based inhibition of an enzyme, and you also had induction of that enzyme, you could then model with the mechanistic static model and include both of those terms and offset that. The draft from 2017 said, no, you have to consider those separately because there's concern for either overprediction or underprediction, depending on which results you get. So they're saying it can be done, and there's concern with concurrent predictions. And if you read more closely, you'll see that you actually have to run the simulations both ways to make sure that you're not missing anything. So another one, this is on downregulation. So if concentration-dependent downregulation is observed in vitro and is not attributable to cytotoxicity, additional in vitro or in vivo studies may be needed to understand the potential clinical consequences. And this can be a very tricky spot to be in because the response for concentration-dependent downregulation may actually be due to the mechanism of action of the drug. There could be other reasons that we don't know about. And when it comes to cytotoxicity, I received a comment from another viewer from the webinar this morning, which is certainly something I completely agree with. Typical cytotoxicity markers, so for example, LDH release, are not always sensitive to some types of cytotoxicity, so it can be quite difficult to rule out cytotoxicity with certain mechanisms of cytotox. So it could be that you have to look at other endpoints. You could use tests that include Alamar Blue, azamulin. There are other, tests that you can use in vitro prior to running a study to examine cytotoxicity or to rule out cytotoxicity for the downregulation. For transporter interactions, there really is not much in the way of change of scope. It's mostly nuance with some exception. The interpretation of intestinal PGP and BCRP, it now specifies the use of IC50 or KI, which is interesting to me because for CYPs, it was now you could use IC50, you didn't have to use KI, and this is the other way around. But if a metabolite of the drug is an inhibitor or the investigational drug is administered by route other than oral, in vivo inhibition of PGP or BCRP may occur if the I1 over IC50 or the KI is greater than or equal to 0.1, where the I1 is the Cmax of the metabolite or the inhibitor drug. So this, I think, is going back to some of the older guidance, other papers that have been published in the past, and we're coming back to that in the final 2020 guidance from FDA. So for OATPs, again, this is not much of a change, but sponsors may now consider adding a pre-incubation step as part of assay validation when determining IC50 values for an investigational drug. So in other words, the pre-incubation time is no longer specified. It was 30 minutes, but there have been examples where either more or less time of pre-incubation are required for that effect to occur. And the R value equation to interpret inhibition is slightly modified, but the cutoff is the same at greater than 1.1. And if you want to see more discussion of how these equations differ between the guidance documents, there's a letter from Andrew Parkinson to Drug Metabolism and Disposition last year. And mates, this is the most substantial change. The inhibition cutoff value has changed from 0.02 to greater than or equal to 0.1, like the other renal transporters. So timing of in vitro studies from FDA guidance, you kind of have to work backwards, really, from the final FDA clinical guidance. And the question is, when are DDI results needed? And if you look at the letter of the clinical guidance, it's before administration to patients. And I know that if you're going into healthy volunteers, this may not necessarily apply, but when you get to a phase 2A, or if you're going into phase 1 in patients like an oncology setting, the FDA states that inadequate studies of DDIs can hinder the FDA's ability to determine the benefits and risks of a drug and result in restrictive labeling, which is probably the most important point, post-marketing requirements or commitments, and or delayed approval. And so I've seen, and this actually quote goes along with that restrictive labeling. So collect enough DDI information to prevent patients from being unnecessarily excluded from any clinical study because of their concomitant medication use. So I've seen more than one occasion where a client is going in, these are mainly oncology space, going into phase 1 in patients, and the FDA reviews the clinical protocol and suggests that they need to include all sorts of restrictions because of COMEDs. And so they have a long list of transporter substrates and inhibitors and say, you don't have any in vitro data, so therefore you have to exclude any patients that are on these COMEDs. So for timing in the PMDA guidance, this is a little bit less direct than the FDA guidance, but it's still early. And usually, this is the final guidance wording in English. Usually prior to the initiation of phase 1 studies, the information of drug metabolism of the investigational drug should be obtained based on in vitro studies. So to me, that says prior to the initiation of phase 1, so you're talking about IND, you need to know a little bit about the metabolism. So is this simply which metabolites are forming? And this can tell you is it metabolized? Do you need to look at reaction phenotyping? Which enzymes are involved? So if you look at in vitro drug-drug interactions in the PMDA, they say usually before the initiation of phase 3 studies. And this reminded me really of the figure adapted from Donald Tweedy, ITC paper from 2013, where the definitive in vitro DDI data, even at that time, were being moved from proof of concept all the way to new drug application, back to first in human proof of concept to discovery to first in human in IND enabling studies. So you have to consider the indication, of course, COMEDs, and other aspects of your program, and really what amount of de-risking do you want to have involved in your program. So these next tables, I'm going to go through these pretty quickly. I'm going to point out my convention here is that additions to the final FDA 2020 guidance will be in bold, red, underline. Differences in the 2018 PMD guideline are just simply in red. And so if you look at, this is for evaluating test drugs as victims and the enzymes of interest. And they are essentially the same for all three agencies, especially FDA and PMDA. They have a first and second tier for P450 enzymes. 2013 EMA guidance specifies test systems, not necessarily enzymes in particular. They do mention CYP and UGT enzymes are present in all systems mentioned, and I'll cover a little bit more detail on this in a later slide. But if you look at other drug metabolizing enzymes, so as I mentioned before, FDA has added carboxyl esterases. They've added back aldehyde oxidase, which I think was a relief to many of us, and they've added the sulfaltransferases. If you look at PMDA, they've included this since their draft, the last draft and now the final, which is dihydropyrimidine dehydrogenase, or DPD. It's not named as such, but is certainly strongly implied by reference to the cerebidine 5-fluorouracil DDI, which caused some fatalities. PMDA also mentions certain UGTs 1A1 and 2B7 by name, and they have some other text within there that shows other enzymes systems should be examined on occasion. So for transporters of interest, really there's not much that has changed in scope. If you look at across the board for which ones are substrates of these transporters, we're talking about mainly intestinal efflux, PGP, BCRP, hepatic uptake with the OATPs, and then of course renal transporters. The only really phrase that has changed in the final FDA guidance is to consider the drug's physiological properties. And this can be important when you consider some drugs, although they didn't name OCT1 by name in the guidance, there are certain drugs that will tend to be OCT1 substrates. And there could be substrates for other transporters that are also not named in the guidance. So that is important to do right off the bat, really, and know your compound and its physicochemical properties. I won't go through the rest of the slide, really, just to note that FDA notes that for most investigational drugs for PGP and BCRP, unless they're BCS class 1, and they really can't be considered that unless the 2017 biowaiver guidance is followed, so there aren't very many drugs in that scenario. PMDA does mention other transporters to consider include OCT1 and MRP2, and EMA considers OCT1 in another part of the guidance. So for a simplified interpretation of substrate potential, this is basically trying to look at what was similar between FDA and PMDA alone. For the intestinal efflux, we're looking at net flux or efflux ratio greater than 2 and significantly inhibited by one or more known inhibitors. Very similar for uptake, greater than 2-fold for hepatic uptake transporters. And for renal, again, significant uptake and inhibition by one or more known inhibitors. So interpretation of substrate potential, the FDA says if positive, consider clinical studies based on the safety margin, likely co-medications that are known inhibitors of the transporters, et cetera. And it refers to clinical guidance for specific clinical designs and also to the FDA website and you'll have this link in there to see which clinical inhibitors they mention. The PMDA is a little bit more nuanced. They, for instance, for PGP, consider GI absorption, brain distribution, and risk of CNS toxicity and renal secretion. And then if the substrate, so this is the fraction absorbed times the fraction escaping gut metabolism, is greater than 80%, we can presume there's no interaction in the gut. And I think that's a reasonable assumption. With BCRP, there is a high rate of polymorphisms in Japan. And so this has a little more attention in Japan because of this. They do note that it is currently difficult to design DDI studies using in vivo inhibitors, but you do need to include that in the label. So for other transporters, clinically relevant inhibitors are listed in the guideline as opposed to the website. So this is a final list. It may be that they update it at some point in the future. For 2013 EMA, they note that when a candidate transporter has been identified, an in vivo study with a strong inhibitor is recommended. So for drug metabolizing enzyme inhibition scope, again, the P450s are essentially the same across all three guidance documents. There are no other drug metabolizing enzymes that I could find within the final 2020 document that were listed specifically for FDA. PMDA lists UGT and 2B7 and others, and that note is that if a test drug is directly glucuronidated, then tests for inhibition of 1A1 and 2B7 and other UGT enzymes, namely those that directly glucuronidate the test drug, which implies that you need to know which UGTs are metabolizing that drug, and you would have to do a UGT reaction phenotyping study first. And you can base that largely on the structure of the compound. I mean, if you have a compound that has a nice phenolic group and you know that it's rapidly glucuronidated or sulfated, then you know which enzymes to be looking at. The EMA has a quote on UGT 1A1 and 2B7 and to study inhibition of UGTs known to be involved in drug interactions. So the note here is that investigational drug is mainly metabolized by an enzyme not listed above. It is recommended to study the inhibitory effect on that specific enzyme if it's feasible. Interpretation of reversible hepatic CYP inhibition. So again, I won't belabor these equations. These are the R values that I was talking about earlier. I will mention that, I should go ahead and put the note down there. The change in the FDA final guidance is that the unbound KI is listed, and the cutoff values are essentially the same across the board. Even for EMA, it's just simply missing the 1+ factor. I would note that, again, FDA cites the paper that allows us to use KI values calculated as IC50/2 when your substrate is equal to Km. I'd also note that PMDA and EMA recommend estimating the unbound inhibitor concentration in vitro due to nonspecific binding, but that's not included explicitly in the equations. So for CYP inhibition, does IC50/2 really equal to Ki when the substrate equals Km? The answer is yes, based on data from 343 experimentally determined Ki values, where we also had experimentally determined IC50 values. They do in fact correlate with the predicted Ki values from IC50/2 when the Km is equal to, or sorry, when the substrate is equal to Km. And as I mentioned, it's now cited in the final 2020 FDA guidance, and this is based on the Ching-Proofsoff equation from 1973. So if you look at the graph that we published, and this is the log of Ki observed, and the log of the predicted Ki, which is IC50/2 on the y-axis, and that correlation is 0.94. I would note that if you're trying to find this reference in the FDA guidance, it's misspelled as HAPUT, not HOPT. So keep that in mind if you're looking for that reference. So the interpretation of reversible inhibition of intestinal CYP3A enzymes, again, the only explicit change that I found in the final guidance was that Ki unbound is listed in the equation. Again, PMDA and EMDA do recommend estimating the unbound inhibitor in vitro due to nonspecific binding, but it's not included explicitly in the equations. And if you'll look at these equations in more detail, they appear to be different, but essentially they are the same, and they have the same cutoff. Even for EMA, that greater than 10 is the same as greater than 11. It's just not, it doesn't include that 1+ factor. So for the interpretation of irreversible inhibition of hepatic enzymes, again, I won't belabor these equations. This is the one where you have the rate of inactivation over the concentration that causes half of that maximal rate. The only explicit change I could find in the 2020 final guidance from FDA was that it now lists the unbound KI value. But again, all these cutoff values are the same. And I would note that here for EMA, that the inhibitor concentration is defined as 0.1 times the dose in 250 mils. And again, PMDA and EMA do recommend in the text estimating the unbound inhibitor concentration in vitro due to nonspecific binding, but again, not included in the equations. So for irreversible inhibition of intestinal, again, as I mentioned, there still is not one. There's no R2 gut in the final FDA guidance. So we don't know if whether or not the unbound or total concentration should be used, or if we should completely ignore that when we have irreversible inactivation of CYP3A4 and a decent gut concentration. For PMDA, they have this equation with this 0.1 factor in there, and the cutoff value then is greater than 1.25. If we had a scenario like this where we had irreversible inhibition of CYP3A4, I might actually consider using the PMDA's equation for the FDA submission, unless we find out that there is a very good reason why FDA did not include this. And again, EMA does not include that 0.1 directly in the equation, so it would appear to be more conservative. But going back, if you look at this, inhibitor concentration really is defined earlier in the text as 0.1 times the dose in 250 mils. So interpretation of CYP induction, this is the basic R3 model, and there are 3 other basic models now in the FDA guidance. And the only change I could find anywhere in here was that the Cmax is now steady state. Everything else is essentially the same. The cutoff values are less than 0.8. This again is for the basic model. These equations may look a little bit different, but they're talking about the same things with the Imax unbound versus the I here. EMA does describe an R3 type equation, but it's not as a standalone static model with its own cutoff value. For transporter inhibition, as far as scope, there really are no changes here. I would say that the FDA does recommend using in vivo index substrates for in vitro assays due to some substrate-dependent inhibition that has been noticed in the past, or to use a probe substrate that provides a lower IC50 for known inhibitors. And this way you get more to the worst-case scenario. If you have a more sensitive substrate for your transporter assay, then you know that you have a better chance of picking up a signal. I would also note that EMA does include OCT1 for hepatic uptake. They do include BSEP as a hepatotoxicity marker. So they do have 2 other transporters, but that's been there since 2013. However, there have been more papers, even the recent ITC paper showing that OCT1 may be a transporter that is of emerging interest. In fact, there are other transporters that may be of emerging interest, such as OAT2, but that's for another talk, another day. So for intestinal PGP and BCRP, these equation and cutoffs, again, these I won't belabor the point, except that I will note that the one change I could find is that for the FDA, it now says you can use IGUT over IC50 or KI. The cutoff values are the same for the intestinal concentrations. Even here, the cutoff for EMA is 10 if the dose over 250 mils is used. FDA 2020 did note that for a non-orally administered drug or a metabolite, which makes sense because it's not being given orally, the equation for that should be I1 over IC50 or KI is greater than 0.1. The OATPs, again, the equations are essentially the same as they were in the draft. PMDA does imply that you should use the blood concentration. So if you look at the difference here, in FDA, we've got fraction unbound in plasma, and in PMDA, you have fraction unbound in blood. The unbound inlet concentration is what used, and we know from the earlier slides that the text may actually refer to the unbound concentration, although I don't know that specifically for the OATPs. One point I want to make here, though, is that the use of the blood to plasma ratio, using the I-Inmax equation for the FDA guidance, it's implied in the PMDA guidance because it uses the term fraction unbound in blood. And this can be very important for certain drugs that either bind to carbonic anhydrase or are actually excluded from red blood cells. So you do have to take this into account for many drugs. And for discussion of that term and how it's used between the guidance documents, there's a letter from Andrew Parkinson that was published in Drug Metabolism and Disposition last year. For the OATs, so these are the renal and mates. Again, the major change here is that the cutoff for mates was increased to greater than 0.1. The cutoff for PMDA is greater than 1.1, which is equivalent to FDA, but again, they've kept the cutoff for mates at 1.02, which is equivalent to the old cutoff in the draft of 0.02. And that also is true with the EMA 2013, where it's equivalent to the PMDA cutoff of 1.02 for the mates. So how do you go about choosing the relevant in vitro concentrations? So use the known or predicted clinical PK parameters if you have them, right? Maximum unbound hepatic inlet concentration. So this equation is simply an expanded form of the equation used to calculate the IH, which is in figure 7 of the final 2020 FDA in vitro DDI guidance. And all this really is doing is coming up with that plasma inlet concentration unbound. You take the fraction unbound in plasma, multiply it by the plasma I-Inmax. You take the fraction absorbed times the fraction escaping gut metabolism, and then the rate of absorption, which is assumed to be 0.1 per minute, which is rapid absorption, the dose in molar over the blood flow, and then the blood to plasma ratio. So I'm sure at this point, many people are asking the question, what if we don't have these data? So one way to do this, if you have selective conservative global target in vitro concentrations. So say you have the unbound IC50 and you want these at the substrate equal to Km, and you have some information about the Cmax. You go 100 times that. To get to the IC90 to fully define that curve, it's simply 1000. For the gut, and this is including CYP3A4, PGP, and BCRP inhibition, you would use simply 0.2 times the I gut concentration over fraction unbound in the incubation. And to reach that IC90, you go 2 times that. For CYP induction, you're pretty much stuck with the limit of aqueous solubility and or cytotoxicity. There's no way to really predict that. But if the unbound plasma Cmax or dose is not known, the limit of aqueous solubility or cytotoxicity probably applies to most of these scenarios, certainly when you're pre-IND. So harmonization. The final 2020 FDA and 2018 PMD guidance often match the final 2013 EMA. And when the PMDA does differ from the FDA, it seems like it still matches the 2013 EMA guidance in several respects. It's somewhat more conservative, as I mentioned, in some of the cutoffs for the mates, for instance. And then there are other aspects. So PMDA and EMA do cover interactions caused by food, alcohol, antacids, supplements. And EMA also encourages in vitro studies with herbals. So to conclude, and then I have a few final comments at the end. The final 2020 FDA in vitro guidance does not have many substantial changes from the draft 2017 document. It's often a matter of nuance, so that should be a relief for many of us that are in this field and don't have to completely redesign studies. The 2013 final EMA guideline is under revision, and it has been for a while, but at this time, 3 agencies do appear to be substantially harmonized in many cases. And then due to the requirement that has come out more from the FDA and PMDA for earlier in vitro data, the concentration ranges in experiments may simply need to be based on limits of solubility and/or cytotoxicity rather than known Cmax or maximum hepatic inlet concentration or the oral dose. So final thoughts. I think you should follow guidance, even if it says on every page of the FDA guidance you don't have to, certainly within reason. And the reason for that is that you want your communication and your correspondence with FDA to go as smoothly as possible. And it does represent the FDA's current thinking, especially now that it's finalized. And there are alternatives though. And so if you look at the example of PGP and BCRP for inhibition assessment, this is a quote from the guidance: the sponsor may calibrate its internal in vitro systems with known inhibitors and non-inhibitors and actually propose a different cutoff value with proper justification. So if you have a large data set, you can come up with your own cutoff values. Basically, the message is that good science and communication with agencies can be a trump card. And so if you look again at the guidance, the language of the guidance, it says these 2 final guidances describe a systematic risk-based approach to assessing the DDI potential of investigational drugs and making recommendations to mitigate DDIs. So you may not always have to follow the absolute letter of the guidance, and it's important to essentially use good science, use good reasoning, and take every opportunity you can to communicate with the FDA and other agencies and come up with a plan that makes sense for your program. If you have an orphan drug, if you have another drug for unmet medical need, there's also the ICH-M3, which has a clause about drugs being developed for essentially fatal conditions, whether that be cancer or other dangerous and unmet medical needs. You can potentially defer some of these studies. So these guidance documents, as it points out, is a risk-based approach, and it depends on the amount of de-risking that you want and can apply to your program at the time that you're going through applications for IND and later on. So finally, where to find the regulatory guidance. You'll have these slides and you can have the links here, of course. And with that, I thank you all for your attention, and we'll take any questions.
Thank you, Brian. While we wait for questions, I wanted to mention that information about our services and test systems, published papers, posters, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Brian's presentation or any of Seksui XenoTech's contract research services or products, you can also ask them in the post-webinar survey or contact us through our website.